Hello, Mr. Congressman. Today I'm going to present you our findings on the CRISPR-Cas9 system, which is a genome editing system, and specifically how it relates to plants. However, before delving into its potential, let me first present some background information. The CRISPR-Cas9 system is still relatively new in science, having only been around since the mid-2000s. It was discovered when scientists realized that many bacteria have repetitive DNA sequences in their genomes, which later became known as clustered, regularly interspersed, short palindromic repeats, or, put more simply, CRISPRs. These repeats are indicated on the image below by the black, unhighlighted portions of the DNA that I'm going to circle right now. Among these repeated sequences, scientists noticed unique sequences, as indicated by the colored boxes that I'm going to point some arrows at. These unique sequences were termed spacer DNA. At their discovery, it was noticed that these sequences were often similar to those found in viruses, and this gave the first indication that perhaps these CRISPR regions were more than they appeared. Along with this, scientists began noticing genes that were associated with the CRISPR sequences. These genes became known as CRISPR-associated, or Cas genes, which I'm circling right now, and are indicated below by the white highlighted region. Through further studies, these Cas genes have been shown to mostly encode for the endonucleases, and these aid in cutting DNA. One such Cas gene is the Cas9 gene, which encodes for an enzyme that has become of particular interest to researchers due to its ability to generate double-stranded breaks. Because of the presence of viral DNA, as previously mentioned, it is thought that CRISPR is a required immune system in bacteria that allows sequences to be integrated from viruses to protect the cell from an infection. It does this through an interaction of two biological macromolecules, Cas9 and guide RNA, which is also called cRNA. Taking a closer look at the Cas9 protein, we have discovered that it is an RNA-guided, double-stranded DNA nuclease. This is part of what gives it such great specificity, and thus where its power really lies. It's able to do this through the interaction of multiple domains, such as the REC1 domain, which I'm going to highlight right now, to which the guide RNA is bound. The helix bridge is then able to bind the target DNA, and the PAM interacting domain is actually part of what makes this so specific. After this has all occurred, the REC2 and the HNH2 bands are then able to cut the DNA. Overall, this system gives bacteria an opportunity to fend off infections by viruses by recruiting this Cas9 complex to cut the viral DNA, and thus preventing the spread of infections. However, we're now at a point where we've been able to take this naturally existing system and modify it to aid our own advances in science and technology. One of the ways in which we have done this is create guide RNAs that allow us to target specific sequences in the genome. Along with this, there are other pathways that can be used to activate the CRISPR response, such as the use of tracer RNA. Overall, there are three basic steps involved in the CRISPR genome editing system: adaptation, cRNA biogenesis, and interference. By intervening and modifying these steps differently, scientists are now able to change the genomes in monocots and dicots with great specificity. As previously alluded to, This new system is proving incredibly important to potential research in plant biology by providing an avenue to create mutant strains, as well as by creating very specific edits in genes of interest. This allows for novel testing with greater specificity than was allowed previously, which ultimately helps us move forward in learning more about genes, their specific functions, and overall, our basic knowledge on plants. Along with this, the CRISPR-Cas9 system is becoming increasingly important to agricultural development. It's no secret that we're at a point where the world is slowly running out of food, and where we're even running out of land to grow it on. And as such, we need to start developing these cutting-edge tools if we're going to try to keep on moving forward fast enough. Right now, our team believes that the CRISPR-Cas9 system offers this by allowing genes to be edited very specifically and even introduced into plants with such precision that there might be a noticeable difference in plant production potential within one generation. Along with this, these new genes being introduced offer the potential to reduce the use of strong chemicals, such as pesticides in day-to-day agriculture. Of course, this has been a lot of information, and I'm sure that now you're left wondering, why exactly might this be important to you? Well, as I said, this technology is really going to be essential in our biological and agricultural development. However, in order for this to happen, my team has determined that we need your help in 3 basic areas. First of these is funding. As a growing area in research, there's still a lot more that needs to be done. And in order to do this, we need some more funding to get this sort of basic groundwork out of the way. Along with this, it would be helpful if an emphasis was placed in education, especially on the development of these new crops, and what exactly it is that goes behind them. Thirdly, a change in policy helping make genetically modified organisms more marketable would help decrease research costs and increase the potential use of this technology, as if people become more aware, then it's also likely that people will start to eat GMOs, and there's going to have an increased demand for them. As it was shown, none of this is actually unnatural, and we were simply using a tool offered by nature to speed things up a little bit. Now, before we conclude the presentation and head out, I would like to ask you just one more question. How many steps are involved in the CRISPR system? And if you're feeling really up to the challenge, what are these steps? Thank you again for meeting with us, Mr. Congressman, and I hope that you found our presentation on the CRISPR-Cas9 system and how it pertains to plants and agriculture both informative and enjoyable.